So the presentation today is about using chemical engineering to help children with cancer. This is something I'm particularly passionate about and something I'm involved with myself, and I thought I'll share that story with you so that you can see how engineering really connects to the benefits of others and to the lives of in this case, a very vulnerable group of our society. So just a little bit about myself. As you heard before from Mark, I am Stefan Guldin. I'm an associate professor here in the department, and I lead a research group that's called the Adaptive and Responsive Nanomaterials Group. In the department, I'm also deputy head, and I am the lead for enterprise activities of our department. So I try to, advise the department on strategy on how to best work with industry, and make sure that the knowledge we generate here in our department is being transferred, to the outer world and to the practice, to the engineering practice that is conducted in companies and in products and so on. My background, I studied physics in Germany, in Karlsruhe and in Munich, and then I conducted a PhD at the University of Cambridge. I then was working as a postdoctoral fellow at the Swiss University called Polytechnique Fédérale de Lausanne, EPFL. And then since 2015, I'm now an academic working as a group leader here at UCL. So the research mission of my group really is to exploit principles of molecular self-assembly to create adaptive and responsive nanomaterials for healthcare and the environment. And I will explain you in more detail what that means over the next 20 or 30 minutes or so. Okay, so this here is just an overview of scale. Okay, so you see here basically a scale bar, and in contrast to, say, a ruler, where we normally use millimeters and centimeters, we use nanometers here. Okay, and so as we can see, we're starting off on the angstrom level, so below nanometer, which is the length scale of small molecules, so glucose, sugar. And then here we have the length scale of macroscopic objects, so a tennis ball. Okay, and nanoscale really is obviously much smaller than a tennis ball, and much smaller than a grain of salt, and also much smaller than a cell in our body. And here you can see how these different objects scale. Okay, so what you can see is nano coming from 10 to the negative 9 is basically the length scale where many processes in biology and also in our body operate. So here we can see a protein is roughly on the length scale of a nanometer. DNA is roughly on the length scale of a nanometer. And then a virus is typically on the length scale of 100 nanometers. So that would be 10 times, 100 times, probably around 1,000 times smaller than the length scale of a cell. And it would be 10, 100, 1,000, 10,000, 100,000, 1 million times smaller than the length scale of a tennis ball. Now, I want to know from you guys, what is the length scale of a human hair? So would you be able to put that in perspective here on that chart? Again, if you could just put down your guess. I will keep you anonymous, obviously, as I said before. But what do you think is the length scale of a human hair? 1 micrometer, okay. So micrometer would be 10 to the power of 3 nanometers, right? So that would be here. I think that's a valid guess. Do you think we can still see a micrometer with our naked eye? Because we can actually see a hair with our naked eye, right? Any more thoughts? Okay, so there is one here, 10 to the power of 6 nanometers, 100 microns. Okay, so we are now going from, we are now in this regime here. So it's bigger. We can definitely see a grain of salt. I think that all sounds quite reasonable. So let's check where we are getting to. And indeed, the length scale of a human hair is around 100 microns. Okay, so that's 10 to the power of 5 nanometers. Interestingly, though, our hair actually grows with a speed of 1 nanometer per second. So you see, again, we are having the nanoscale, even in our hairs, when we look at the speed of the growth of the human hair. Okay, so in my group, we are really interested in nanoscale science because we want to design materials that can effectively interact with these biological entities. We want to design materials that can detect proteins, that can recognize DNA, that can kill viruses. And so we need to have this kind of structural control. We need to have the right size, and we need to be able to tune interactions between our materials and those biological entities. And some examples I want to show to you today. Okay, so the first example, why nano? So why do we need to control materials on the nanometer length scale is shown here. So what we can see here is just a surface. So that's basically a glass window. And what happens is, if light shines on a glass window, we all see reflections, right? So a part of the light is being reflected. And there are certain rules of physics that would tell you exactly how much of the light is being reflected, which is encoded in the Fresnel equations. Okay, so what we want to achieve in anti-reflective coatings is that we cancel out reflection, and this is already done. So if you buy glasses, at your local optician, you will have the option of having an anti-reflective coating. And these operate as you can see here. So they will have a thin coating on top of your glass, which basically is designed in a way as such that light reflected on the top of that coating and light reflected on the bottom of the coating is being cancelled out. And as a result, the light is able to penetrate fully into your substrate. Now, there are certain equations that help you to design such a coating. So you would then know, for example, what is the refractive index, which is a material property. What does that need to be for this coating, and also what does the thickness of your coating need to be in order to get this kind of cancellation? Because what you need is phase matching, so that basically the phase of your wave, the minima and the maxima, is exactly aligned so that they cancel each other out, and also the amplitude of your wave, so the height of your wave, the strength, intensity, these need to be matched. And so there are certain equations that help you to guide your materials design. Now, why nano? Well, basically visible light is based on wavelengths that are on the nanometer scale. Okay, so here you can see, across the visible spectrum, blue light is a smaller wavelength, around 400 nanometer, red light, larger wavelengths, around 650 or so nanometers. And what that means is now that if you want to have a coating that is not recognized by the light, that is below the resolution limit of that incoming wavelength, you need to be much smaller than that wavelength. As such, then your coating becomes, seen as an effective medium, and these kind of pores, for example, here that are used to tune the refractive index, they are no longer recognized as something of a feature. They're just going to be looked at as a mixture of pores and of inorganic material. And so now, if you therefore need to make features that are so small, much smaller than the wavelength of light, then you do need to use molecular self-assembly. And this is what we do here. So what you can see is basically in this approach, we use a molecule, a macromolecule called a polymer, which has two separate types of blocks that are covalently linked. Now those two blocks here demonstrated with two separate colors, they dislike each other. So they would form, certain nanoscale structures to obey the laws of thermodynamics. And you could now introduce some extra material that associates with one of your two blocks. And what you then do is you are structuring, you're basically structure directing, in this case, inorganic material using organic structure directing agents, these kind of polymers. And this is then molecular self-assembly that is happening. And what you can then do is you can, after your processing, remove the organics, and you end up with a porous inorganic film where the structural features are directly related to the self-assembly that was imposed on it. And what can then happen is that you are first obeying those laws of anti-reflection, but also because you've now structured your material on the nanoscale, you are able to include some very potent material, which in this case is titania. And this titania normally would be very reflective, but because it's now structured on the nanoscale, you can include it, and it can be used to make this kind of coating self-cleaning. So as you can see here, this is a coating that was put into where basically a fingerprint was applied and put into the sun for an hour or so. And on the left-hand side, the substrate without coating, the fingerprint was still there. Whereas on the right-hand side, with the coating, you could see that this fingerprint was completely degraded or removed. Another example is shown here. Why nano? Because we can make tough materials from sustainable sources. This here is so-called abalone nacre. So abalone nacre, if you look on Wikipedia, is described as being a unique material that is extremely tough and structurally resistant. And why is that the case? Well, because it combines organic and inorganic materials. You could make a very hard material, but often that material would be brittle. You can make a very elastic material, but that material wouldn't be tough, right? So the beauty of abalone nacre is that it's a combination of both inorganic and organic material, and it's arranged in this kind of fashion on the nanometer scale. So you see that you have these inorganic calcium carbonate platelets that give you basically the hardness, but then these platelets are connected in a matrix of organic matter, and this organic matter is like a glue, and provides you now with with its strength that this material is not just brittle. Okay, so it's really a beautiful material to be inspired from as a materials engineer. And actually, if you check Wikipedia for abalone nacre, you will see that in 2012, researchers created a calcium-based nacre in the lab by mimicking its natural growth process, and you would find my name there because this is what I have been doing during my PhD. And this was basically the outcome that we published at the time. So you can see, we've been trying to learn from nature. We've been trying to find a way to also create an organic matrix where these inorganic platelets can grow through. And as a result, we basically had a material that was extremely structurally resilient and can be used for quite interesting applications because it's also biocompatible. And as said, it can be manufactured from sustainable sources, so with low energy and low environmental impact. Okay, so this is the last example of why I think nano is interesting and it's important. This is a nanoparticle. So you see here on the nanometer length scale under a transmission electron microscope, you see these kind of dots. So these are little spheres of gold, and we can see them with the naked eye because, again, they resonate with a certain wavelength of light. So in this case, you know, they are having an absorbance peak in the red, and this therefore allows you, you know, to basically see the color of these dots. Now, what you can see is that the absorbance of the particle changes when they are getting in close vicinity. And this is what is demonstrated here. So you see in the first example, basically we have the nanoparticles in an organic solution down here, which is dichloromethane. And then on top here, this is water. So if we shake this, what's happening is that the particles get to the water interface and they are getting hydrolyzed. And then when they're hydrolyzed, they are reactive, they're becoming reactive. And when they see another particle, they are starting to stick together, just like Velcro. And therefore, what happens is that you've kind of forced them to form you know, a membrane, to form a layer by hydrolyzing them, by making them reactive. And this process can be used, and it's kind of showcased here, to make a very thin layer of gold nanoparticles. Now, in this particular example, I just wanted to demonstrate how nanoparticles change their optical properties, and that we can see this with the naked eye. But we can also make a sensor based on this principle by, for example, forcing those nanoparticles to stick to a certain cancer biomarker or to a certain virus, and by kind of agglomerating in solution, then changing their color. Okay, so I want to kind of go back to the original mission of this presentation, of how now chemical engineering and nanoscience can be used to help children with cancer. So in the UK, there are currently around 20,000 children with cancer, living with cancer. And many of them, in fact, over 50%, get a certain type of medication, which is called anthracyclines. These anthracyclines are really, really toxic. And so you have to be very careful in giving the right amount of medicine to children, okay? And the challenge now comes from this little cartoon down here. Every child distributes this medication in a different way. Every child metabolizes this medication in a different way, and every child clears this medication in a different way. And currently what doctors do is they don't take this personalized response into account. They simply measure the patient and look at what is their body surface area, what is their weight, in order to then determine the right dosage for the therapy. And this leads to quite some problems, which is shown here. So if we have 3 patients, as I said before, these 3 patients react in a different way to chemotherapeutic drugs because they have a different clearance, different metabolism. And what then happens is that, you know, patient A, in this case, would be underexposed because the medication is cleared very quickly. Or patient B would be, you know, in the sweet spot because, you know, this patient behaves exactly as the models predict that are based on body surface area and weight. And then we have other patients where, you know, they don't clear the drug as much. It stays in the body for much longer. And the body therefore, you know, has severe adverse effects. And so actually what you can see is in many chemotherapies, children develop later heart problems because of excessive exposure. And so really there is a need to determine these personalized parameters carefully to assess the concentration of the drug inside the body. Because if you know what is the concentration of the drug inside the body, you know what is the profile of that patient and how that patient needs to be treated as such that the medication is in the sweet spot. Okay. And so actually this is currently being done in some model studies, which is called therapeutic drug monitoring. The issue is that it's really complex to do that. So basically, you have to draw the blood of the patient. You have to then process that blood. You then have to send that blood into a centralized lab, and that blood has then to be analyzed. And then we have to feed back this data to the doctors so they can make the decisions. And this all takes often up to 3 days. And so really what you want is you want to have a point of care analysis. So you want to basically, right at the bedside of this patient, determine what is the concentration of that drug, as such that you can directly respond and adopt that treatment and make it the best for that individual patient. So we were looking in my lab at novel methods to be able to do that, okay? And so here is one example. We were designing nanoparticles that, when they had none of that drug, none of the, in this case, doxorubicin associated around them, they would be soluble in one liquid. And then when they basically had receptors, we trained these nanoparticles such that they would be selective for this type of chemotherapy drug, for doxorubicin. So what had happened is if doxorubicin was present, the particles would bind it. And once they bound the drug, they would then go into the complementary liquid. They would undergo a phase transfer. And this is shown here with some images and here on the cartoon. So basically by measuring the extent of phase transfer, we were able to basically measure the concentration of doxorubicin in the bodily fluid. And this was then basically implemented into some sort of prototype shown here. So in this kind of large European project with various companies and universities and university hospitals, we were basically building a prototype that was based on nanoparticle phase transfer. And this allowed us to, to assess the concentration of drugs in blood, in this kind of device architecture. Now, unfortunately, this was really complex, and we realized that it's not trivial to basically implement such a device or integrate such a device on the ward, because of the way, because of the types of solvents it needed, because of the time it took, because of the skills that were needed to maintain this equipment. So we're looking for a much simpler solution. And actually, there is a solution available that I think many of you have already seen before. So I just quickly want to show this to you now. So here I have a filter paper. Okay, so this is similar to a coffee filter paper, and I made a few dots on the bottom of this filter paper, not on the bottom, but a little bit further up. And I'll put this now into this beaker where the water is below the position of those dots, and that will make the water creep up the filter paper, and then we're going to see what's going to happen. Do you guys know what kind of principle this is? I'm sure quite a few of you will have basically had some experience with that type of experiment, in your school, right? So, what is that called? Do you know? Can we have some comments in the chat? Okay, so that's chromatography. In fact, this is thin layer chromatography. Okay, so I'm holding this paper now in the water, and basically the water creeps up that paper. So I can show you where we are right now. Basically we can see that there is already some color separation, and I will leave it now inside and let it run, because it takes some time for the water to basically creep up that thin layer, and we will look at this later on then and see where we got with this experiment. Okay, so basically we were using thin layer chromatography for the detection of chemotherapeutic drugs, And this, is quite intriguing because it's very simple, but you need to obviously do, some tweaks to make this a quantitative process. And so I want to show this video now, which basically provides some of the principles of what we call quantitative thin layer chromatography.
Hi, my name is Stefan Guldin, and I'm the head of the Adaptive and Responsive Nanomaterials Group here at University College London. Today we are launching our web app QTLC. This is a freely available tool that allows you to perform quantitative analytical chemistry with your smartphone.
Thin layer chromatography, TLC, is a simple but powerful method that enables us to physically separate the chemicals in a solution. This is often used in a quantitative context, but did you know that TLC can also be used for quantification? Our QTLC app makes this easy, but how does it work? You need to prepare a TLC plate by spotting a small but defined quantity of liquid a short distance from the bottom of the plate. To use QTLC for quantification purposes, you need a calibration curve in addition to the liquid you want to test. On this side, you spot known concentrations of the target molecule, your calibration curve. And on this side, you spot the sample with an unknown concentration. After running the TLC plate, you can take a photo and upload it directly to the QTLC web app. Inside the web app, you can define which molecular bands you are interested in. The web app then computes the relative number of molecules within each band based upon their shape and intensity. You can then plot the data using any data analysis software to obtain the concentration of your analyte.
Thank you for joining us today. We hope this will become a useful platform for teaching and learning. A full paper with student handouts, experimental plans, and detailed instructions are provided via the displayed link. We welcome your feedback and comments.
Okay, so this is just the principle of quantitative thin layer chromatography. So we've basically built this app as such that you can perform simple experiments, and I showed you just here that I was basically using even coffee filter paper. You can perform whatever experiments you want to do for your quantification purposes, in your school laboratory or in your university laboratory or wherever it's safe to basically conduct this experiment. And then you can take a photo of your plate, and that may be either in absorbance mode, so you just take a photo, outside with the adequate background, or it may be in fluorescence mode when you basically have to excite the molecules with a certain wavelength and then take a photo. And this is basically shown here, this difference. So then you would need to kind of take a photo through a fluorescence box. And then we will provide you with the data as such that you can then plot your results and then basically determine the concentration of the unknown sample. And basically, this app has been, has been quite widely used. So far we can see users from over 37 countries across 6 continents. And we've also won the UCL Education Award 2020, and we've been covered in various media outlets, and now being kind of recommended by the Royal Society of Chemistry for implementation in basically school lessons and so on and so forth. Now, this kind of technique can also be used in the quantification of chemotherapeutic drugs, and this is shown here. Okay, so as I said before, a very common therapy involves the drug doxorubicin. And what you want to see to understand the metabolism of the patient is also how the patient is turning doxorubicin into doxorubicinol, which is basically a metabolite. And so what you can see here on the left is this approach of having calibration curves to guide the concentration of the unknown concentration of the compound. And we can see here the separation, how basically thin layer chromatography has enabled us to separate the different compounds that can then be quantified, individually. So up here is epirubicin, which is a very similar compound to DOX. You can see it's really just, this feature here that is different. As a result, it's traveling very similarly to doxorubicin, and there's just a small separation of the band shown here. And then you see here doxorubicinol is a bit behind. And this is because doxorubicin has here this ketone, and here we have this alcohol group. So otherwise, again, these molecules are very similar. And that's so unique and fascinating that with thin layer chromatography, if you use the right mobile phase, so the right solvent to travel across your plate, you can really nicely separate different compounds. Okay, so basically, if you use our app, you can now quantify the concentration of an unknown amount of this drug. And what we have shown is that we can do this even from patient samples, and that we are, as you can see here, very reliably within a 15% error margin, which is what we need, where we need to be for approval by the regulatory authorities. And what we also saw was that we can actually achieve this level of accuracy with very, very small blood volumes, so down to 5 microliter of blood, which is interesting because it means that you don't have to take a huge blood draw. You could also just use a finger prick sample. Okay, so the Chromadose solution is basically shown here. We are now trying to turn this principle into a functioning device that can be used on the wall. So this would need to be housed in an automated desktop unit. We would need to do this kind of chemistry in a safe fashion. So we need to have this in a cartridge that is completely sealed. And then we need to have smart analytical algorithms to be extremely precise based on those images that we acquire. And actually, we've gone already quite a long way. So we have, we have basically filed a patent for this novel idea. We've, also widely spread this principle using the QTLC app and also some other activities. We have published a number of papers on this front to also demonstrate that what we do, is widely recognized and accepted by other researchers. And then also we've demonstrated the necessary performance for this to become a viable solution. So how would this Chromadose device work? Well, basically, all the principles would need to be housed in this box. And then, the nurse would take a drop of blood, would basically feed this into the device. The device does all the processing based on the principles as shown before, and then gives a very clear recommendation. So in this case, we would need to reduce the dose by 10%, for example. And we would do this over time so that basically, doctors get a continuous response and potentially are able to fully understand the response, the physical response of this patient. Okay, so now, going from what works in the lab with the manual operation to a fully tested device that is safe to use on a hospital ward is really complex, right? So you have to go through many, many stages of engineering and work with many, many different experts. But ultimately, we learn these kind of skills here at UCL because a lot of this boils down to the engineering design cycle, which is shown here, where you're going in an iterative fashion around this kind of structure, that is there to help, to guide you, where you go from the need to the brief, to the conceive phase, the testing phase, the instruction and specification phase, the make phase, the testing phase, the use phase, and then potentially, through further iterations until you finally have a product that really is fit for purpose. So in this project, for example, we are working with a very large and interdisciplinary team. I myself, the lead of the project, together with Professor Gareth Wheel in Newcastle. But actually, we are not only working with engineers, we're working with lots of nurses, with doctors, we're working with parents, we're working with children, with designers, with consultants. We're working with a whole broad spectrum of people to make sure that this technology is going to be safe to use. It's going to be widely accepted and hopefully also powerful in its performance. Along the way, we've also founded this company, Visynta, which is the commercial vehicle now to produce this type of product. So it's a university spin-out that is now independent and employs currently 5 people. And basically altogether, we have the vision of making this technology a reality. Okay, so I'm at the end of my presentation. I thought before I finish off, I just want to say why I personally think that UCL is a great place to be. It's a great place for students. In our ethos, we take very great pride of making a connection between research and education. And this has been phrased as the UCL Connects the Curriculum. So basically, in many of our lectures, we are implementing, novel approaches that come out of our own research and the research of others. We're giving you the opportunity to participate in these research questions. And also, we're giving you the opportunity to listen, for example, to cutting edge research carried out in industry, from startups to blue chip companies. We're having scientists visiting our lectures and so on and so forth. And actually over 2/3 of all modules in UCL Chemical Engineering have this kind of approach. So have direct participation from both research and industry. We have this integrated engineering program at UCL, which means that, we're not just building or educating different types of engineers in separate programs. We are making sure that all these engineering disciplines, civil engineering, biochemical engineering, biomedical engineering, chemical engineering, electronic engineering, mechanical engineering, computer science, and also management science, that these have joint modules, cross-cutting modules, which allow you to work in more interdisciplinary teams, which allow you to understand what it takes, to be efficient in a team. And also, we are providing you with additional soft skills and also scenario-based learning, experiential learning, to make sure that you will become, a new generation of engineers that can solve the problems of the future. And one example is also the integration of a minor. So this is kind of a side subject along your main engineering discipline, where you could take a minor in applied chemistry and molecular engineering, which is the one that I'm, for example, leading. And this would then allow you, for example, to learn more about applied chemistry and molecular engineering and to tackle some of the questions I've elucidated today on. And this would be possible irrespective of whether you are a chemical engineer or a biochemical engineer or an electronic engineer, because all of these types of engineers could take this minor and kind of excel in this research direction. Okay, so I'm at the end of my talk today. I want to acknowledge my group shown here. I want to acknowledge our funders, but most of all, I want to acknowledge you. Thank you for your interest, and thank you for your interest in my research and also your interest in UCL as a whole. And I'm looking now forward to the discussion we may have via the group chat and also the Q&A option that you have via Zoom. Thank you. Okay, so there's a first question around, can this technology be used in drug concentration testing in, say, athletics, for instance? So it depends what type of substances you want to detect. So at the moment, the technology is very competent or very powerful in detecting fluorescent compounds. So, for example, we've been also implementing this technology for the detection of antidepressants and also antifungals, antibiotics, and so on. So there are many drugs where this technology is directly applicable. But if your compound is not fluorescent, we would need to tweak it. We would need to either tag it with a fluorescence marker, or find another way of making it visible for our system. And so that is the challenge there. So I'm not saying that this kind of principle is applicable to all types of drugs, but to many it is already now, and we're trying to make it applicable to more. I'm actually working with a startup that is measuring hormone levels in sweat with a different type of technology. So, it's a very agile field. And again, our expertise is needed, or is at least, is at least useful. So I would say nanoscience can help you in many ways. It's not always the principles of the linear chromatography though. Okay, so there is another question here. I learned at school that there are different types of solvents to use while doing chromatography, especially acid or methanol. Why do we use water here? Okay, so that's a very good question. So basically, I'm just using water in this particular example because I'm sitting in my office and I need to be working in a safe environment, right? And I'm only trying to separate out these colors. So now here you can see kind of how they've been separated. Okay, so you can see different color bands coming from obviously one type of pen that we're using in the past. So we've been separating the colors into their, into their original components that were used in the dyes. Now, the separation here on this plate is actually not as good as what you could see in the videos, right? So this is because we're not using the right mobile phase, and we're not using a very powerful substrate. So it's because we're just doing this with a filter paper. So now the comment is exactly right. In order to get a better separation, in order to get a very tight separation as such that we can do this in a quantitative fashion, we would need to tune exactly the composition of your mobile phase. And that is tuning the acidity, tuning the polarity. So there are different handles, and you need to adjust this very carefully as such that whatever is the mobile phase, that it's directly applicable to the types of molecules you want to separate. And in fact, you wouldn't be able to separate doxorubicin from doxorubicinol if your mobile phase wouldn't be perfectly tuned for this type of application. So thanks for this comment here. Any more? Any more questions? Just put them in the Q&A box. Okay, so the next one is, do you think that egg yolk can be a good outer membrane for fabricating a stimuli-sensitive nanocomposite in cancer treatment? Okay, so egg yolk as such is amphiphilic. Okay, so amphiphilic means that so egg yolk is protein, it's a macromolecule. One end of egg yolk is more water loving, and the other one is more water hating. Okay, we call this hydrophilic and hydrophobic. Okay, so in that sense, egg yolk is very useful. In fact, we're using egg yolk in mayonnaise, right, to stabilize oil droplets. So definitely it can be used in a formulation where you want to encapsulate a material that is otherwise not very soluble. So the question is it the best material that can be used? And the challenge here is, if you introduce a medicine in the body, it will be already directly exposed to various, it will be exposed to attack mechanisms from your body. There will be proteins forming around your object that you're introducing. And the question is, how does now your nano shuttle or your nano carrier that carries the drug respond to those mechanisms, those response mechanisms of the body? And so I would say we have meanwhile found better ways of performing drug delivery. So I don't think that egg yolk is the most powerful or most potent way of doing that, but it's definitely, from the principle, suitable. So yeah. Okay, so there's a question here from an entrepreneurial standpoint. Can you tell us efforts you've made to make this product scalable and ultimately affordable for low-income communities? So it's a bit tricky to develop a medical technology. I mean, this one is not going into the body, but it's still used to guide decisions, very important decisions. How much medication do you give to a particularly vulnerable patient, right? And so you have to comply with the strictest of regulation in your development. So, you know, like you have to write a lot of documentation around your development. This documentation has to be checked by the authorities. And you have to obviously go through many, many cycles and then implement this in clinical studies and so on and so forth. So what I can say is that we are still in this process. So we are not even in the application of this technology in the clinic, where we would need to do clinical trials and so on. So it's still a few years down the line. I think the beauty of this technology is, though, that it's not extremely complex. As I showed you before, you can actually achieve very high accuracies even with your smartphone by looking at the separation of compounds. So I would think that it's very well suited to be implemented in also developing and low-income communities. But before we are concerned about the scaling and about you know, bringing it to more applications and more patients, we need to make sure that it's working in a safe way and reliable fashion. So this is our current emphasis. Okay, and then we have another question. How do you decide which type of engineering you should pick for your degree? Well, I think you can go from two different angles. You would, I mean, if you're sure that you want to be an engineer, then either you can go from the end of your involvement. So you could check what are the types of products that this kind of discipline is involved. You know, do I like the surrounding? Do I like to work in teams that are dealing with this kind of question? So that would be going from the back end, I would say. Or you think from the front end, you know, looking at the curriculum, you know, how do I like these kind of modules? You know, what is most suited for my interests? But I would strongly advise that you do both, so that you're not only looking at the curriculum to make a decision which of the type of engineering you want to do, because ultimately, once you've graduated, obviously you'll be evolving in that discipline, and you know, every undergraduate course will have some somewhat dry fundamental modules that you will have to take to just learn the basic skills, and then it will definitely become more and more interesting as you advance through the curriculum, because you're suddenly learning how to apply your skills to more tangible questions. So in that sense, I think I would look at it from both ends. You can also engage with the various societies, for example, the Institute of Chemical Engineering and other types of learned societies for the different disciplines. They will have events for young engineers that will help you decide whether this type of discipline is the right thing for you. But I'll definitely be open and also talk to people that have the experience. All right, if there's no more questions, then I think we could end the session now. Please do reach out to me. You know, if you having another question about this talk later on, you will find my website and my contact email by simply googling me, Stefan Guldin. There's only one that you can see on Google, and just reach out to me. I would say it was a very stimulating Q&A session. I enjoyed presenting some of the principles of my work to you, and I hope this was also relevant to you and that you found this an enjoyable experience. Yeah, would be great. to see you in person one day if you decide to come to UCL. Please, stop by. We also have open days. We have outreach events. There are lots of ways of engaging with us, and hopefully, I see you again. And for now, all the best to South London, to SLO, to Zimbabwe, and to Pakistan. And yeah, hope you're having a nice day. Bye.